Hello, NeuroMatch. I'm Jordan Matelsky, a PhD student in the coding lab at the University of Pennsylvania. Today, I'm going to talk about the connectome, the wiring diagram of the brain. And I'm going to share some exciting early results about how we used some information from other biological processes to see how the connectome is constructed. We want to ask the question, how is the brain wired? What are the rules that determine how a neuron connects to other neurons? These are some of the fundamental questions of neuroscience, and an enormous amount of research has gone into answering them. We know that cells change as they divide during the development and the lifetime of an animal, and it's generally understood that they differentiate into cell types, different classes of cells with similar phenotypes. Often these cell types are used to build predictive models of how different neurons connect to each other. On the right side, I'm showing an adjacency matrix of my toy connectome with 8 neurons, and you can see that the cells either connect within the same cell type, these are the colored boxes here, or to other cell types, the rest of the matrix. For example, I can build a probability distribution that says that all neurons of the red type synapse on blue neurons with a 4% probability. Now, when I want to know if this one particular red neuron and this one particular blue neuron share a synapse, I simply flip a 25-sided coin. And in fact, that's not only how I know if these two neurons connect, it's also how I know if any of the red and any of the blue neurons connect. Everything in the yellow region has a similar probability of connection, since those boxes represent the synapses between the same neuron types, that is, red synapsing on blue. But this makes a pretty serious assumption. Many of these models imagine that all neurons of the same cell type have the same connectivity behavior. And it's not controversial to say that we know that's not true. It's just a convenient model because the brain is so complex. But can we do better if we use other streams of knowledge? Well, let's look at what we know about the brain. We know that all cells in the body are related. So we can ask the question, how distantly related are two neurons A and B? Here I'm showing a dendrogram of all of the neurons in the brain with the zygote cell at the top. And each time the cell splits and forms two daughter cells, I can represent that with a fork in this tree. For example, if we want to know how distantly related the red neuron and the blue neuron are, we can simply count the number of hops. So we go 1, 2, 3. So we can assign a lineage distance of 3 to this pair of neurons. And now we can combine that adjacency matrix from earlier with our new dendrogram data sources. Here's our new combined data set. We have both lineage information, which is the dendrogram on the sides, and the connectome information, which is the adjacency matrix in the middle. Now let's see what kinds of questions we can ask now. Let's look at these 3 cells, 1 green type and 2 blue types. If we ignore the dendrogram model for a second, we would imagine that this middle blue neuron here probably behaves most similarly to this bottom blue neuron, since they are, after all, of the same cell type. But when we look at their actual connectivity, we see that this blue neuron actually has more synapsing characteristics in common with this green neuron. In other words, the distance between the middle blue neuron and the green neuron is 2, and the distance between that middle neuron and the bottom blue neuron is 3. That is, that middle blue neuron is actually more closely related to the green neuron, even though it's a blue cell type, and its network connectivity reflects that fact. In fact, if we completely do away with cell type information, we can still see the same effect, totally ignoring the cell types that we've worked so hard for years to classify. Here's that same adjacency matrix where we didn't even bother to add cell type information. All we have is the cell lineage data, and we can still predict an extraordinary amount of the connectome. We applied this technique to the C. elegans nematode, a worm with a few hundred neurons in its whole body. This animal is a great model organism because we not only have the complete reconstruction of every neuron and its chemical synaptic connectome, we also have a complete full-body cell lineage family tree. So let's ask the question, if I know a neuron's connectivity, does that tell me anything about its siblings' connectivity? So we'll take all pairs of 2 neurons and we'll look at their lineage distance and their similarity in terms of the connections. So we might imagine 2 ways in which these properties could be related. We might imagine the plot on the left, where closely related neurons have more partners in common. This trend suggests that similar neurons have similar wiring, so if I'm friends with you, then my brother is more likely to be friends with you. Or conversely, we might imagine the plot on the right, where if I'm friends with you, my brother is less likely to be friends with you. In other words, we would expect this if closely related neurons maximally diversify their local connectivity neighborhood. And in fact, because the C. elegans connectome is so stereotyped in between individuals, we can ask the same question for a bunch of different animals and see if the effect still exists once you account for the natural variance of the connectome. And it does. We learned that for all of the connectomes we examined, more closely related cells in the cell lineage had more downstream connections in common. In other words, the more closely related two neurons are, the more likely they are to share connectivity characteristics, whether or not they're the same cell type. It's pretty common to think that neurons that are physically closer to each other tend to be more similar in their connectivity patterns. And one thing that I think that is really exciting about this work is that the effect is actually amplified if you control for the physical geometric distance between the neural cell bodies. Now, there's obviously a ton of science that I don't have time to talk about today, but what I want to communicate is this. If you know where I synapse, you know a lot about where my sister synapses, even if my sister and I are different cell types. And if you know where I synapse, you know very little about where my 10th cousin is going to synapse, even if my 10th cousin and I are the same cell type. Now, our hope is that this equips us to start to unravel the connectivity of the brain through the lens of simpler proxies. If we can learn the connectivity patterns of a set of representative neurons, then perhaps we'll be able to predict the connectivity of their closely related cousin cells without having to closely study every single cell in the brain. And we also wonder if violations of this rule might actually point a spotlight at particularly important or noteworthy connections, or maybe even pathological connections that we otherwise would have completely missed. Now, perhaps C. elegans is too small of a connectome or too unique of a body plan, and so our intention is to generalize this to other model organisms. New connectomics data sets such as Genelia's hemibrain and new genetic marking techniques like BRIC-seq from the Zador lab may pave the way to explore this relationship in larger organisms, either in subvolumes or across entire brain areas. All of our work here is online, shared as open source and reproducible Jupyter notebooks on GitHub. So with a single click at that link up there, you can jump into a live notebook and start to explore our analyses, or you can bring your own. We're actively looking for data sets that co-register lineage information with connectome graphs, and we're really excited to collaborate, so please do reach out. I'm looking forward to hearing your comments and questions. Thank you.